Hello students, welcome to my channel. So in this class we will see about the site-directed mutagenesis. As we see the name site-directed mutagenesis, what does it means? Site means at a specific place. Mutagenesis means कुछ न कुछ म्यूटेशन करेंगे वहां पे. जो नॉर्मल वाइल्ड टाइप DNA we take and then we do mutation in that DNA sequence. And what topics we will cover? First we will understand what is the site-directed mutagenesis, and then two major topics are in it: non-PCR based method which is doing mutagenesis, and one is PCR based method. And in non-PCR based method we learn about these three topics: cassette mutagenesis, design of mutagenesis, and primer extension mutagenesis. Okay, first we will see the what is site-directed mutagenesis. Site-directed mutagenesis is an in vitro method, means we are doing in the laboratory for creating specific mutation in a known sequence. We know already the sequence where we are doing the mutation. So known sequence will be reform or modify, and which will convert into the mutation and non-wild, non-natural form. It is fundamental tool widely used in molecular biology and protein engineering. In molecular biology, we majorly consider nucleic acids, and protein engineering, that's why we consider is a differently. But mostly it is also considered as molecular biology. Allow the researcher to design selective changes in DNA. Means what? In this mutagenesis, we allow scientist or any researcher to do any type of mutation or change in DNA. That is the experimental biology, and provide powerful method for probe gene regulation, how gene will be get regulated by our humans, and relationship between protein structure and their function. So why this is powerful method? Because we can know the gene regulation, how gene is getting regulated and how we can modify it, and which kind of modification give which kind of result, what kind of result, and what is the relationship between structure and their function. If something is have same molecular basis in some DNA, but protein, if there is in protein there is 100 amino acids, and these 100 amino acids, same to same amino acids are present in different type of a protein. This one is one protein, this is second protein, but the structure of this protein is different from this protein. So we see that these two proteins are functioning differently. Then how this is happen? So we can understand that type of relations, the structure and function, relation between structure and function. Function and structure role of amino acid residue can be studied by the comparing. So we can compare and can know what is the functional and structural role of any amino acid by comparing mutant protein with the wild type protein. And how we know the mutant protein? We will do the changes in protein sequences. Intentionally we do that type of thing. Conventional mutagenesis means what is the traditional method? What is the classical, or at the starting of this technique, what type of method is used to mutagen or do the mutation in some molecular biology? That time the mutation was random process, that is not the site specific process, and introduce changes at unspecified position because we don't know. We do randomly mutation in anywhere in DNA sequence. But nowadays we are doing site-specific mutagenesis. Require screening of large number of mutant organisms to find the desired mutation. And what is the problem with that? If we do random mutation, we have to do screening of whole genome or whole DNA sequence which is used in that particular mutagenesis. So it has large amount of organisms genome. So this will take extra energy, extra processing details. So to avoid this, we have a site-specific mutation, and the sequence we know where we done the mutation. So this is make simple and easier to identify and less time taking. Then to find out our desired where our non-sequence is. Then site-directed mutagenesis. Now we are doing site-directed mutagenesis. How? Compared to natural or random mutation, this is best because mutation are constructed by genetic engineering. We are doing so much research and then we are doing this kind of mutation. It do not require extensive screening, and this will save our time. Extensive screening and process of doing working on single thing will decrease. Type of in vitro site-directed mutagenesis, as we already discussed, which is PCR based and non-PCR based. Two type of site-specific mutagenesis, which is one is PCR based and second one is non-PCR based, in which we are not using PCR tool. PCR based are more frequent. It is more frequent, more efficient technique or more efficient method to do the site-specific mutagenesis than the non-PCR based. But we discuss how the non-PCR based technique is best site-directed mutagenesis is done.